Good evening, everybody, and welcome to the Italian Culture Institute in Tokyo for this fifth edition of a Day of Italian Research in the World. This evening will be moderated by Professor Enrico Traversa of the Embassy of Italy in Tokyo. And I have now the honor to give the floor to the Ambassador of Italy to Japan, Mr. Gianluigi Benedetti.
Buonasera a tutti, good evening. Sorry for a little delay. Dear Alberto, thank you very much. It's always a pleasure and nice occasion to be here in this wonderful conference room. Dear speakers, dear participants, it is with the extraordinary pleasure that I welcome you at the Italian Institute of Culture in Tokyo for our first scientific event in person after the beginning of the pandemic. Today we are celebrating the Italian Research Day in Japan, an initiative promoted by Italian Ministry of Foreign Affairs and International Cooperation, together with the Italian Ministry of University and Research, with the interesting seminar, Italian Brains in Japan, Italian Researchers Studying Neurosciences in Japan. The human brain is the most fascinating and mysterious organ, and we still fail to understand the typically human behaviors such as consciousness, learning, or memory, making the field of neuroscience one of the most appealing scientific fields that needs a multidisciplinary approach. Science, technology, and innovation are top priorities for our vision for developing the relationship between Japan and Italy. And amongst the various initiatives, our embassy is promoting the visibility of the Italian talents who work in Japan. And we became aware of the significant number of Italian researchers who are at the forefront of neuroscience research here in Japan. Therefore, we decided to display their innovative research activities with this seminar, organized in collaboration with the Association of the Italian Researchers in Japan as a portion of the Italian Samurai Initiative. Under this initiative, Italian Samurai, we are posting online on our social media videos of Italian researchers working in Japanese institutions in science, technology, engineering, and mathematics fields. And I thank very much Professor Sara Cosentino, President of the Association, for their collaboration. We believe that those small clips can definitely attract the curiosity and the interest of many other talents, even maybe younger talents in Italy, and that will eventually create a much larger mobility from Italy to Japan. The presence of Italian researchers in Japan is very important because they are testimonials of the high quality of Italian science and research, but most of all, because often, and I would say always, they are the bridges, the real bridges, for the promotion of collaborations between Japan and Italy. I'm therefore looking forward to hearing our talented speakers tonight, sharing their knowledge, and I am sure that this webinar will contribute to fostering collaboration between Italy and Japan in the field of neuroscience. Please enjoy the webinar. I wish you a very fruitful discussion. I am very curious myself, and a very stimulating learning experience. Thank you very much.
Thank you very much. I call to the floor Sara Cosentino for a brief greetings to the audience. Thanks.
Thank you very much for your kind introduction. Good evening, everyone. I'm Sara Cosentino, the president of the AIRJ, the Association of Italian Researchers in Japan. So I spent just 2 words on the association. The association was founded in 2019. So after that, the pandemic began, and we don't have a lot of activities in the history. But we started the association for a variety of reasons. One reason, of course, is to foster the community of the Italian researchers in Japan, so for social purposes, for cross-pollination, and various scientific activities. Also, the idea is to acquire funding that are possibly acquired by programs of collaboration between Japan and Italy. For example, we are now working on establishing the JSPS Alumni Italy Association, which is, of course, another association for JSPS alumni, but it will come with a few perks for us, and also with a couple of scholarships, possibly, in addition to the ones that are for Italian researchers in Japan. And so I don't want to take too much time, but you should look up our website, airj.info, for the information, and maybe if you want to join the association. Thank you very much. Good evening.
Thank you, Sara. And now we are ready for starting for the talks. The first talk will be by Dalila Burin of Tohoku University. And so please, Dalila, the floor is yours.
Thanks very much, Professor Enrico Traversa, and thanks to the ambassador for inviting me today among the Italian brains in Japan. We all study the brain and the nervous system, but we do it from very different perspectives. In my personal case, the focus of my research is the bodily self. I study how the brain represents the body and its movement in normal and pathological conditions, and how to exploit those processes for rehabilitation purposes. You have probably seen already this picture, this weird homunculus here. This is actually its definition. The brain literally has a map of the body on the surface of each hemisphere contralaterally. But this map, though, does not perfectly represent the body as we see it. But body parts that are more sensitive and more flexible take up more space on the cortex. For example, the fingers are relatively small, but they are extremely sensitive and extremely flexible, so they occupy quite a big space. And this is the result. This is literally how our brain sees our body. What is interesting for our purposes is that this body schema is modular, is composed of several components and subcomponents. It's very plastic. Think of, for example, when you use a tool, the body schema kind of updates to temporarily integrate the tool within it. And crucially, for my research activity, it can be altered, it can be manipulated. How? Essentially in 2 ways. With a direct damage in the brain that can be a stroke, a hemorrhage, or a traumatic event, or with experimental manipulations, especially multisensory illusions. One of the most innovative, compelling, and recent multisensory illusions that comes directly from the entertainment industry and became my favorite research method in the last years is immersive virtual reality. Immersive virtual reality essentially is a computer-based world that can reproduce or simulate the physical world. What is important for me is that in this virtual world, we can have a virtual body that can completely replace and overlap the real one. Imagine if you could wear a visor and you would look down towards yourself, you would see your virtual body displayed coherently with your own. And that would be enough to create the illusion of ownership over this virtual body and its movements. Now, I'm sure you're all thinking, how is this even possible? How can anyone in full control of its mental faculties believe that this is such a strong illusion? And the answer is quite simple, because the world is virtual. What we call world, what we call reality, is nothing but the reconstruction that our brain does of our surroundings, starting from what we perceive thanks to our senses. So maybe now you're thinking, if there is no difference between physical world and virtual reality, why do we need it? What are the advantages of the virtual reality? Well, in virtual reality, we can do everything you can imagine, and even more, because virtual reality surpasses the physical limits that, of course, the world necessarily imposes. In the virtual world, for example, we can have different somatic features. For example, my skin is white, but my avatar can have a black skin. Or we could be children again, and just like kids do, we could perceive objects as bigger than their actual dimensions. In fact, the embodiment of the virtual body can have several consequences on the real body on different levels. And that's precisely what I studied in my research, in my career so far. First, the movements of the virtual body can affect my own movements. This is actually one of the very first studies I did with immersive virtual reality. If the virtual body performs movements different from the ones I am doing, for example, if I am drawing straight vertical lines, as you can see here now, but at the same time my virtual body performs ellipses, I will adapt my motor performance, because the illusion of having this virtual body is so strong that I will change my movements to match them with the seen one, with the virtual ones, even if it is in contrast with my own motor intention. Clearly, this is an effect that can be exploited for rehabilitation purposes. And that's what we did in this study in collaboration with my colleagues from University of Turin. We assessed a group of patients, neurological patients, mostly with a neurological disorder in the left hemisphere of vascular nature. And we assessed them with a battery of tests for several motor functions, especially for the lower limbs, including speed, endurance, gait, balance, and so on. Before and after a virtual training in two conditions. In the experimental condition, the virtual body was displayed in first-person perspective. So, as I said before, overlapping the real one and creating the illusion of owning this virtual body. And in third-person perspective. So, basically, the virtual body was displayed in front of them. So, it's like looking at someone else. We found an improvement in some of the assessment tests, specifically after the training in first-person perspective only, so when the training is performed with the own virtual body, and specifically all of those tests regarding the balance domain. So we can actually say that this virtual training was effective for balance, for the improvement of balance in those patients. As for the rehabilitation of the upper limbs, and specifically for the hands, together with my colleagues from Politecnico of Turin, we are developing an engineering glove that can record several parameters at the same time, such as wrist rotation, strength of pinching, of grasping, and some physiological parameters, to be used, of course, with neurological patients and in combination with virtual reality. Actually, this is a project financed by the Italian Ministry of Foreign Affairs within the executive program of scientific and technological exchange between Italy and Japan. Unfortunately, because of the pandemic, the project is delayed, but it's ongoing. So the motor effects are probably the most intuitive that we can explore and we can exploit with virtual reality. But what about other consequences? What about, for example, physiological effects? In this study, this is actually one of the very first studies I did when I came to Tohoku University, and that's financed by a JSPS grant for early career scientists. I was saying the training, the virtual training in this experiment is similar to the one I described before. So basically the participants is still sitting on the chair, but the virtual body performs a high intensity intermittent exercise. Basically, the avatar switched from running to walking every 30 seconds for a total of 8 minutes. During the training, we measured the heart rate, and we found, as you can see in the graph here, that the heart rate goes up and down coherently with the virtual movements, even though the subjects were completely still sitting on the chair. But this happens only when the training is performed in first-person perspective, so with the own virtual body. In the same study, we also measured for potential cognitive effects, because this is exactly what happens after a real physical exercise. So before and after the same training I described before, we tested the participants with a very famous test for cognitive executive functions called the Stroop task. You probably have seen it. It's the one where you have the name of a color written with a different color, and you have to say the color of the ink, not the name, not the word itself. So for example, blue written in green, you have to say green, not blue, as quickly as possible. We found, again, that after the training in first-person perspective, the response time decreases. So participants were actually faster. There was actually an improvement in their cognitive performance. Oops. But if this is not enough to convince you, we also measured directly the brain activity with this device here called functional near-infrared spectroscopy, FNIRS for friends. And we measured the hemodynamic cortical changes over the prefrontal cortex, which is considered mainly the brain localization for the executive functions. And we found specifically an increased activation over the left dorsolateral prefrontal cortex, and even more specifically, a peak activation a few seconds after the display of the stimulus during the Stroop test, again, only after the training in first-person perspective. So we actually found a direct correlation with the brain activity after the virtual training in first-person perspective. For the record, we replicated this same study on a group of elderly, and we confirmed all of those results, but in a long-term perspective. In the study with the young participants, the training was composed of only one shot, only one session, but with the elderly, we found the very same results in the long term, after a longer training. Last but not least, if a virtual training can have beneficial consequences comparable to the ones that arise after a real physical activity, what other effects can we exploit? And there I realized stress. There might be an effect on psychosocial stress response and on subjective levels of anxiety after a virtual training. Again, in a similar protocol, as I described before, the subjects are sitting on the chair still and looking at the virtual body, running for 30 minutes at 6.4 kilometers an hour, if I remember correctly. We measured before and after the training the salivary alpha amylase, which is a very common, very famous biomarker for stress response, and subjective levels of anxiety. We found actually that the salivary alpha amylase decreased after the training, the virtual training in first-person perspective, together with the state level of anxiety. So this training, this virtual training, was actually effective in reducing the stress response in the acute effect. Concluding, we have seen how it's possible through appropriate experimental manipulations to trick the brain and to transfer beneficial effects from the virtual body to the real one on several levels, from behavioral to neural. I also thought for the purposes of this meeting was interesting to briefly introduce some of the collaborative projects together with the Italian universities and research centers. I hope it was all interesting and curious, and perhaps there will be some more time to discuss during the panel discussion at the end of this presentation. Thank you all very much.
Thank you very much, Dalila, for this fascinating presentation. Maybe we are going to end all to doing gym virtually. And so now I leave the floor to Marco Terenzio of the Okinawa Institute of Science and Technology. Please, Marco.
Grazie. Thank you for the kind introduction. This is where I work. This is the OIST in Okinawa, and if you have a chance, please come and visit. And our group is interested in neurons. Now, you're probably all familiar with nerve cells, of course. This is an example of a neuron in culture. And what you can notice is that it has this rounded body, it's the cell body, the center of the cell, and it has this huge arborization, which is very extensive. Now, this is a little bit deceptive, though, because if we are looking at the relative proportion of the two compartments, actually more than 1,000-fold, there is a more than 1,000-fold excess in the network compartment than compared to the cell body. In fact, if you think about certain neurons, like the motor neurons, which start from the spine and go all the way to the extremities, those could be more than 1 meter in large mammals, including human. So the neurons we are studying specifically are two kinds of neurons. Let me see if I can put. Okay. So one kind of neuron is this. These are the sensory ganglia, which are outside the spinal cord. And those are sensory neurons that project to the extremities, and they are the sensory of pain, the mechanoreception, and the sense of balance, which is proprioception. And then these are neurons which are resident in the spinal cord, and those are the motor neurons that determine the movement. So the main sort of question that we are trying to address is, given this huge disproportion between the volume of the network and the soma, how can the neuron, the nucleus at the level of the soma, actually even know what's going on in the periphery? And that this is really, really important, because in a lot of neurodegenerative paradigms, the axon compartment, these protrusions, they die, they degenerate a lot sooner than any effect can be seen in the soma. And therefore, this is also applicable to injuries, by the way, including traumatic injuries. So to study axonal biology is really critical to understand the mechanism behind axonal injury repair and also certain type of neurodegeneration. So our unit deals with two main arguments, which I'm going to try to touch a little bit today. One is the investigation of the mechanism that underline axonal degeneration. And the second time, we're trying to devise way to improve regeneration. So in terms of the first sort of line of research, what happens... Sure. Okay. Sorry, I'm not used to speak from a podium, so... all right. So is it better now, the sound? Is it better? Okay. All right. So this is an enormously simplified schematic of a neuron, which is contrary to everything that I told you before, because the axonal compartment is very short. But nevertheless, you can see that there are signals from the periphery. In this case, it's simplified by this sort of cargo thing here. And they go from the periphery all the way to the nucleus, and that's thanks to this all-important retrograde molecular motor that brings them from the periphery to the center. And in that case, in the case of mammalian cells, the major retrograde motor is dynein, at least in axons which are polarized. And this is an example of movement of lysosomes, which are degradative organelles that moves retrogradely along these axons from the periphery to the soma. And one of the things that is really important is that this is actually not just a scientific curiosity, but it's really critical because mutations in the dynein motor that brings these signals is associated with impairment of motor and sensory function, as you can see in this mouse, which has the legs at odd angle phenotype, by which basically the legs are cramped instead of being relaxed as they should be. But that's not true only in mice. This is actually an outdated sort of review of various mutations on the dynein motor. And as you can see, in human, they've actually been associated with neurological defects. And interestingly, what I showed you before, those are mutations that interest, let's say, the biggest part of the dynein mutant, which is the heavy chain, which is this huge blue structure here, which contains the motor domain. And it makes sense because that's the business end of the dynein complex. It contains the motor domain, and it promotes the walking along the microtubules that you see here in green. However, the dynein complex is a supramolecular complex, is formed by a variety of subunits, and today I want to tell you a story about a specific small subunit that is teeny tiny. It's part of it's one of the light chain of dynein, and it's only 11 kilodalton, so 96 amino acids, really tiny. And I was working on this a few years ago, and we had mice in which subunit was deleted genetically. And so we can put this mice to a test. So one of the simplest tests that we can do is you can put a mice and you can put them on a rotating rod, and generally the mice are very good at balancing themselves for an extended period of time. However, these mice in which the subunit of dynein was deleted, they couldn't stay on this apparatus at all. And if you look at the way they walk, you see this is a normal mouse. They walk as you would expect. And this is a mouse in which this small subunit has been deleted. I hope you can appreciate that they don't walk quite as well. And there is a reason for it. And then again, this is another photo of this mice. This is the mutant mouse. And the reason for it is that in the sensory ganglia where the protein was deleted, we are losing roughly 50% of the proprioceptive neurons, which are responsible for the balance. So these mice cannot balance. And turns out that this is due to a defect of retrograde transport of mostly lysosomal organelles. So these are lysosomal organelles moving in the network. And this sort of diagram here, basically what they represent is the movement. So these are frames of a movie, and this slanted line, this is one lysosome that is basically moving from the periphery to the center. You can see that in the wild type, you have a constant movement of these organelles, but if you look at the mutant in which the protein has been deleted, these are straight line down, so the organelle is static, it doesn't move at all. And that's also true, so this is the mutant, and these are various sort of wild-type-ish animal, and you can see that there is a real skew of the curve from a speed that is between 0.5 to 1 micrometer per second to a speed that is below 0.5 micrometer per second. And so this is an example of visualization of lysosomal movement that we can do in a culture where we apply a treatment to the entirety of the culture. However, that's not ideal because ideally what we would like to be able to do is to apply a treatment or a probe to specifically the end of the neurons, so to the axons, because that's why we are interested. And so there is a way that we can do that, and Emily in my lab, she's a PhD student, she has done a wonderful job, and she has been designing microfluidic chambers, and these chambers, the idea here is to separate two compartments fluidically. So we have these very, very tiny microgrooves, and then these microgrooves essentially connect two reservoirs. In one, we put the cells. And in this reservoir, if we have a difference in volume, because they're connected by very, very, very small grooves, they will equilibrate over time eventually, but they equilibrate so slowly that we can do differential treatment. And so, for example, here you can see fluidic separations being maintained up to 24 hours. So if we have two chambers, in one we plate the cells, and in the other one we have axons passing through the grooves, then we can treat the axons and see what happens selectively. And this is an example of this, where she cultures sensory neuron, and these are cell body site. You see a lot of axons there because, of course, they expand axons even though they plate it, but the axons then migrate through the grooves, and this is just axonal compartment with no cell body. And if you put, of course, growth factor to attract the axons, like NGF, then growth factor, and you will have a bigger network. And then we can actually apply a lysosomal probe, for example, to only the cellular compartment and really precisely look at the transport. And in this case, for example, we can calculate the speed very quickly, very, very nicely. We can see that we have lysosomes that go retrogradely with this sort of speed, around 0.7 micron per second. We can see we have a population with a similar speed that is anterograde, but the peak is different because we have less carrier that goes anterogradely from the center to the periphery than we have from the periphery to the center. And then we can calculate how many they are moving. So most of them are static, but there is a significant percentage of carrier that move. And we can calculate how many poses they make. So for example, here we have about 35% of our moving lysosomes, which make 0 pose. Then we have roughly the same percentage, which makes 1 pose, and then it goes from there. And another thing that we can look that is related to transport that we are very interested in is looking at the transport that happens from the nucleus to the periphery. And in that case, this is regulated by the kinesin motors, of which there are several. And the transport that we are interested in is the movement of mRNA in granules associated with the RNA binding protease, this AIRBP, from the nucleus to the periphery. And the reason we are interested in that is because there is an ever-growing cohort of messenger RNA that are being identified in axons. So a messenger RNA are the template that are needed for the creation of the protein, and the protein are the building machine that actually perform functions in the cells. That is an oversimplification of what it is. And why is this interesting? Because we find several neurological diseases, including the amyotrophic lateral sclerosis, where we have mutation of RNA binding proteins such as TDP43 and FUS. And there is a growing cohort of evidence that points at the fact that in this disease there is an accumulation of granule of this RNA binding protein in the soma, which prevents the localization of the mRNA in axon, and that could be connected to their phenotype. I will remind you that this disease, the way, for example, ALA manifests over the year, is that you have a progressive death of the neurite that happens before the soma is affected. So one of the ways the homeostasis of the neurite can be maintained is by having mRNA there, because instead of localizing the protein directly to the axons, you can localize the mRNA, and the mRNA can be translated several times. So you can create very quickly a lot of protein. And so if you think about the movement, anterograde movement, and if you think about a speed that is around 1 micrometer per second on a neuron that is 1 meter long, you can quickly calculate how long will it take from the protein to be transported all the way. And that is an enormous expenditure of energy, and this process makes it a lot cheaper. So so one of our plan is using human pluripotent stem cells from patients of ALS and having a co-culture with the target cells, which are motor neurons, and investigate the process of axonal transport and mRNA localization. So we have a cohort of cells from patients in the lab. They take very, very, very long to differentiate, over 30 days. They start from this indifferentiated state, and then they go through a state of like embryoid body's structure. And then after a long period of differentiation, you have neurons like this, which are acetylcholine positives, and they're bona fide motor neuron from patients of ALS. And then we will use these neurons now to characterize the axonal mRNA translation and localization. And we are also experimenting with co-cultures with muscles to see if we can investigate also the interaction of motor neurons derived from human pluripotent stem cells from ALS patients, and if there are any differences there. And I would like to pass to the second part, which is much shorter, which is ways that we are looking to improve axonal regeneration. So I actually come from a regeneration background. During my postdoc, I was working a model of peripheral regeneration after traumatic injury. One of the things that I would have never expected to do is to do what I'm doing now, which is basically experimenting with 3D scaffold for regeneration. This is because I was very fortunate to have in the lab a nanomaterial engineer, Lokesh, who is very competent. And together we were able to sort of kickstart this line of research in the lab, which I'm still learning. But the reason why I'm interested and we are interested in this is because while the traumatic injury of the peripheral nervous system in many cases can regenerate, depending on the extent of the lesion, when we are looking at spinal cord injury, this is a completely different situation. There's a lot of limiting factor there, including the scar that it's a very good thing because it prevents you from bleeding to death, but it also impairs the regeneration. There aren't really effective ways to treat it just yet, though there are a lot of attempts to do that. And so one of the things that I find fascinating that we can maybe make scaffold that we can implant and possibly one day use in the clinic in combination, for example, with pluripotent stem cells that might actually give a mechanical bridge and support that will help to promote regeneration. And this is very overly simplified schematics, but the idea would be to give a physical support so that the neurons remaining at the other side of the lesion could repopulate the site of lesion and reconnect differently to what they do now, which is mostly doing commissural sprouting, so just going around the site of lesion. So there are different scaffolds that are being typically used. So most commonly, people use 3D fiber scaffold. We also use them in the lab. But today, I actually wanted to talk to you about 3D printed scaffold. This is actually not really 3D printing. It's the opposite of 3D printing. In 3D printing, you're taking a 3D printer and you're actually putting material. In this case, you are taking a drop of a photoresist using a laser to cross-link in the position at the exact geometry that you want, and then you develop your structure by removing the material that is not being cross-linked. So it's kind of the opposite of a 3D printer. But what you can do nanostructure that are really, really precise. We are talking about nanometer size structures. And you can do any geometry you can think, because you can just design the computer and then the printer will do. And so what we decided to do is we decided to try a variety of geometry to see if we can promote vectorial growth of axons. So this is one of the scaffold that we used. You see that we have control region, which are flat, to see how the axons grow on a flat surface, which is a normal flat surface that we use in culture. And we have this grid structure. And in this grid structure, we have smaller nanostructure, which are designed so that the axons cannot really cross over the fiber. They really have to go straight. And the idea is there, can we maximize the growth of an axon? Instead of growing radially, can we push all of its energy to grow in a straight direction? And the answer is yes. So these are axons, are neurons grow on a glass surface, completely flat. These are on the flat surface of our scaffold. And you see that they grow in all directions, right? All the angles. But on the grid, they grow in one direction, very, very, very straight, which is what we wanted, this productive growth from one side to the other. And this is just visualized here. So you have straight lines, and then as soon as they hit the flat surface, they grow in all directions. And this is a 3D model where basically we interlocked the grids so the neurons can actually this time pass through the grid. And so we wanted to see if the neurons can grow inside the scaffold in a 3D way, not just on a planar way from one side to the other. And the answer is yes. So you see here it's color-coded, but it's for the depth. So you have neurons that are on top, and they go and they project axons all the way down, and you can see it better here. This is the top where the neurons are seated, and this is the volume of the axons going all the way through the scaffold. And hopefully this is sort of like a wish, if you want. What we would like is, like in this situation, neurons migrating from outside the scaffold, and you can imagine this scaffold or similar implanted in the site of lesion, and now the remaining neuron migrating through this, and then as soon as they hit the scaffold, going through a straight direction and then quickly go to the other side of the lesion. That's one possibility that we would like to believe is possible. So in summary, I just showed you how defects in axonal transport are really, really important for neural survival, and I gave you an example of a subunit of dynein. I discussed how we can use compartmentalized chamber to separate axons and cell body and study the two compartments separately. I showed you that we can use iPS cells from patients to derive motor neurons and study what happens there. And we can make nanoscaffold. And the last acknowledgement slide, I'm sorry I went a little bit out of time. I want to acknowledge the people in the lab that did the work, so Laurent, Sandra and Sarah for the stem cell work, Lokesh for the scaffold, Maria and Miki for the microfluidic chamber, and the support section, Paolo and Menoa for all the help that they gave us with the project. Sorry, I was 2 minutes late. Thank you so much for listening.
Thank you very much, Marco. And this was an example of how we change topics. I mean, within neuroscience, a totally different approach from to biology and unwillingly materials too. Now we move to the talk of the last talk, Andrea Benucci from the Riken Brain Research Institute.
Okay. Thank you very much, Enrico Traversa and the ambassador, for this event and for the opportunity to present here today. So the rather casual kind of title that I've been using today reflects a trend that we're observing in the neuroscience community in relation, especially to systems and networks neuroscience, to integrate developments in the field of laser technology in artificial neural network machine learning research. This is something that my lab is very interested in, as well as many other labs around the world, and is attracting is producing not only very interesting innovation in the field, but also is attracting a lot of fundings. So broadly speaking, my lab is interested in studying visual perception by integrating experimental and theoretical methodologies, and as I mentioned before, we favor the use of artificial neural networks. This is a sort of a very broad and general statement that is very helpful in sort of redirecting the focus of the kind of research that we're doing in the lab, but it actually conceals a tremendous amount of complexity that we've appreciated a little bit also in the previous talks. So just to give you an idea, when we say that we're interested in studying visual perception in the brain, of course we have a brain, but if we start to zoom in a little bit into the structures, we have, for example, circuits of neurons, and each element in these circuits will be a cell that has very interesting morphologies. And as we keep on zooming in, then we find these elements that are synapses that enable the communication between neurons. And we can keep on digging, going to the protein level, if you want, to the atomic or subatomic level. We can also go to the other side and zoom out a little bit. The brain is not an isolated organ, it sits in a body, and the body physiology has a very strong influence on the dynamics of the brain. And as organisms that interact in a very dynamic environment, we are affected by other people and by what happens in the surroundings. So when we say that we want to study visual perception in the brain, we should be able to define more or less at which scale we want to operate. And one approach could be a bottom-up approach, meaning if you're very ambitious, you can try to start, for example, from the synaptic level and say, well, if I understand the synaptic physiology, perhaps I can understand how the neuron works, and from that I can understand the circuit's dynamics, and so forth and so on. This approach, besides being incredibly ambitious, also suffers of a very profound conceptual problem, which is that there are also a lot of top-down influences, where this scale influences tremendously the smaller scale. And it is also important to mention that at each and every scale, the complexity is further increased by the fact that there are processes that act at different time scales. So not only the spatial scales, but also the temporal scales are extremely diverse. So just to give you an idea of this top-down signaling, Given that the fact that I study visual perception, I'm going to use a visual illusion. Many of you might have seen it already. This is the brick wall illusion. In this brick wall, there is a cigar, a lit cigar, stuck in this wall. Very hard to see if you've never seen the illusion before, but if I highlight the cigar, it's going to become very obvious. Now, what is interesting in this illusion is that you cannot undo it. So from just these few seconds where I was going back and forth with the slides, so just these few seconds of retinal activations of a specific pattern, there has been a change in your perceptual processes so that for the rest of your life, you're going to always see the cigar. It is impossible to undo it, as I mentioned before. So this is just one of many examples of top-down influences across different scales. And in this example, the time scale is very long. As I mentioned before, for the rest of your life, you're going to see the cigar. So this is simply reflecting a pretty obvious observation that the brain is an adaptive system in a continuously changing environment. And so you need to have these top-down effects that act at all spatial scales in your brain. So how do we move forward if we want to try to understand brain functions given this level of complexity? So one possibility is just to give up if you want. Another one is to make great simplifications. And this is what most of us are doing. And one of these great simplifications is to take a time snapshot. So you can try to freeze time and say, well, I don't care how I got to a given point or how the system is going to evolve in the future. I just try to understand brain functions at this moment in time. Or if you want another way to put it, it's like if I had the possibility to decide at this moment in time which kind of information I should extract from your brain, which one is the relevant temporal and spatial scale that I should be looking at in order to understand or possibly reproduce a given brain function. So following this line of making great simplifications, an approach that has been very successful is to use the circuit level scale. More specifically, the idea is that what matters is, to understand a given brain function, is to look at the communication between neurons that exchange with each other electrical signals, like these electrical pulses or action potentials. And they do so, they chatter with each other using this signaling. They do so with different strength or with different weights. So this W stands for weight that connects neuron I and neuron J. So some neurons interact with each other very strongly with strong weights and other more weakly. But according to this very simplifying assumption that we're making here, this is the relevant level of information that you need to gather if you want to understand brain functions. And this is, has been strongly influenced by the fact that more or less these very same mechanisms can produce very powerful computational systems. So in artificial neural networks, you have a very similar framework where an input is activating units instead of neurons. And these units talk to each other with different weights, so influence each other with different strengths, and then they produce an output. And the name of the game here is to fine-tune these weights so that you can produce very specific input-output functions. So all the specialized AI systems that you see nowadays are fundamentally using these sort of operations. And these systems are fully deterministic and controlled by specific equations. So following this very simplifying working assumption, if you want, the underlying idea is that all the rest is what essentially nature had at its disposal in order to adjust these connectivity weights between neurons and enable computations in complex and dynamic environments. As I mentioned before, I want to stress that this is a working hypothesis, okay? So it's something that is possibly going to be disproved. But as a null hypothesis or a simplifying hypothesis, has the advantage that you can take it as far as you can, and when you're going to fail or when you're going to find limitations with this approach, then you can ask, what is the next step? What is the next element that I need to introduce in my working framework to overcome specific obstacles? So far, it has led research at the systems neuroscience level pretty far, and it doesn't seem to have critical limitations, I would say, at this point. Another very important point of this working assumption, working hypothesis, is that it allows to make very strong experimentally testable predictions, which is very important, of course, in science. So today I would like to focus on the idea that in learning new computations or modifying existing ones, what matters, according to this working hypothesis, is to implement specific changes in the connectivity WIJs between these weights that connect neurons. Now, this idea of remodeling the weights of communications between cells has been exploited tremendously in science fiction books, in the popular imaginario, and since my colleague Dalila before introduced the matrix, I want to pick up on this example. So you probably have seen the movie, and in this famous scene, the main character is implanted with this rod, which is going to reconfigure part of the brain, essentially modify these weights of communications between the neurons, and suddenly he could learn new motor skills like Kung Fu. So in a way, what we wanted to achieve was something conceptually very similar, to test the hypothesis that indeed this level of representation is the most relevant one if we want to understand brain computations. So we wanted to test the hypothesis that indeed learning new computation or modifying existing ones can be achieved via changes in the network connectivity via modifications of the weights, interaction weights between neurons. And instead of looking at the model representations like in the movie, we're going to look at visual computations, and we're not going to ask Keanu Reeves, but we're going to use a mouse. Much less glamorous scenario, but probably easier to interpret. So the encoding that we wanted to study is something that is very well defined and has been very well known in the neuroscience community. It's a kind of computations that is implemented in the back part of your brain where the primary visual cortex is located. And over here, if you zoom in a little bit, you will find neurons that have a very characteristic property, meaning they respond very strongly to stimuli that have power at specific orientations. Here I color-coded the preference for different orientations of different neurons. So the yellow neurons will prefer this kind of orientation power. The blue ones will prefer vertical kind of power, and so forth and so on. This is very ubiquitous in mammalian species, in primates. The specifics differ between species. So for example, in the mouse, neurons with very different preferred orientations can sit nearby, but for example in the cat or in the monkeys, they tend to cluster in big patches. And this gentleman here, Hubel and Wiesel, because of this finding and related research, got a Nobel Prize in the 80s. So one characteristic here is that this network of neurons has almost an equal number of cells that are tuned to these different orientations. So another way to put it is that the encoding of stimulus orientation, the computation that I'm interested in this example, is relatively uniform across different stimuli. Here we're looking at the population response of the entire network, but you can think of it as, if you want, the probability of detecting specific stimulus orientations by this network. I'm oversimplifying a lot. In reality, this distribution is not flat. It follows very well the statistics of the natural inputs, but for sake of simplicity today, we can think that this is rather uniform. So the name of the game here, the sort of matrix scenarios that we're trying in a very simplistic way try to reproduce, will be to change this kind of encoding, this kind of computations. Instead of having the network to be a uniform detector of all orientations, we would like to make it a stronger detector of a specific orientation, and do so by modifying the synaptic weights between neurons, so the way the neurons influence each other. So this is the first problem that we need to solve. How do we modify these weights here? Unfortunately, other people before us have been dealing with this problem. So this very famous form of hagioplasticity postulates that if you have two neurons connected to each other through a synapse, it is the relative timing, so the time interval between the presynaptic firing and the postsynaptic firing that determines whether or not this synapse is going to be potentiated. Another way to say it is that neurons that tend to fire together at the same time, so that they're sort of carrying similar information, they wire up together, so they strengthen their communication. The problem is that although this is rather nice and simple conceptually, if you think about a single synapse connecting two neurons, you have a problem when you want to scale this up, because if your population has hundreds of neurons, then you're dealing with tens of thousands of synapses, and you need to control the strengthening of these synapses across these very large numbers. So how do we achieve that? This brings me to the laser part in the title of my presentation, and a lot of innovation that we've been seeing in recent years in this field of optogenetics. So what we have been doing, we have been using a digital micromirror device, which is very similar to the DLP of this projector that we're using here, to project luminance patterns into the primary visual cortex of a mouse. This is not a regular mouse, this is a transgenic mouse that has been injected with some viruses. And the bottom line here, what you need to know is that this construct here called GCAMP allows to identify the activity of a cell by looking at the fluorescence that the cell emits. So if a cell is emitting a lot of fluorescence, it means that it's very active. This other construct here, crimson, is an optogenetic construct that allows you to use a laser pulse to a single cell and activate it. So here's an example where we achieve control of individual cells with fairly good temporal resolutions. And as you can see, even nearby cells can be controlled in isolation. So cell 4 can be activated without any crosstalk with cell 5 and vice versa. More recently, we put this technique a little bit on steroids, I would say, by using holographic techniques, which is a much more improved form of this DMD technology that I mentioned before. I think a video is also the easiest way to show you how these things work. Let me stop here. Okay. So here, when you see a white square on the top left, we're activating the cell that is sitting at the end of the green arrow, as at the same time we're monitoring the whole network. And in these other videos, when you see the white square on the top left, again, we're activating the entire network. And we're also monitoring the activity of this network. All right, so the first observation that we made here is that when we activate with a laser pulse a single cell, we actually produce very widespread activations. So if we look at the probability of exciting cells as a function of distance from this target, we see a decay, but we also see that the probability of excitation continues to be above chance levels, even to the far edges of the imaging windows here. And the other observation that we made is that there is a preference to activate cells that are co-tuned with the targeted one. So here is all color-coded, meaning that if we target cells that has a preference for a specific orientation in blue, then the co-activated cells also have a preference to have the same preferred orientation. This is what this plot indicates here. And so this is all you need to know till this point to understand this paradigm. So in this paradigm, we activated a single cell, and as I just told you, this produces a network-wide activation with a bias for co-tuned cells. Then we waited a short Hebbian time, that's how I would call it, so a little time interval, and then we pulse the entire network with the laser. So as I mentioned before, this little delay of activation in per pulse stimulations is supposed to strengthen the connections between cells that are co-tuned with the target cells, so the blue cells, and all the other connected cells. So the corollary of that, or the expectation, if you want, is that now the whole network should become more responsive to the preferred orientation of the target cells. So this is what we wanted to achieve. We wanted to potentiate the response of the entire population, making the entire population a better encoder, essentially, or a better detector of a specific orientation. So in the sake of interest, I don't have the time to go too much into the details. I'll just go to the result, which is somewhat what we were expecting, but not exactly. So the expectation was to see the population response, the circuit response, to follow these solid blue boxes all the way up here. But instead of observing this kind of response, this is what we got. So we got actually a zero effect exactly at the orientation that we wanted to be potentiated, or that we wanted the network to be better at detecting. So it seemed a little bit of a disappointment at the beginning, but actually turned out to be very trivial. The explanation is pretty obvious and is very simple to say here. So as in the scheme that I showed you before, when we excited a single cell, we also produce activations of cells that were co-tuned. So if the cell was blue, we excite a network of other blue cells, okay? So they have the same preferred orientation. And then when we excited the whole network, again, we excite all cells, including the blue ones. So the blue cells have been essentially excited twice as often as the other cells. And this is very well known to induce what is called synaptic fatigue. So now we have two competing mechanisms, one that is supposed to potentiate the response and one that is suppressing it. So the two cancel each other out. But this phenomena is very, very specific of the blue cells, so cells that have this specific orientation, so it doesn't appear for these other stimulus orientations. And so the idea now is that we have anyway a mechanistic understanding of how the system is going to react to our manipulations. And so if we want to produce an increased detectability of a given stimulus, we just need to take into account this secondary mechanism. But we can still have a control of the network detectability properties and how to manipulate them using lasers, pulses to this network, and transgenic technologies. So then the next question, following again the matrix example, would be whether or not this change in the network that we've been able to induce can also alter perception in the animal. And this is a work in progress where we're leveraging a new setup that we have been developing to train animals with voluntary head fixation. So these animals are training themselves. This is a RICAM sponsor patent. We can train about 48 animals per day, producing 12,000 trials per day. And in a recent paper, we have leveraged the power of these setups using 1.3 million trials to demonstrate specific psychophysical effects in mice. So I think I'm out of time, so will I just summarize? So what I've been showing you today is that there is a strong effort in technology-driven innovation to interrogate, meaning like monitor and excite neuronal circuits down to the single-cell precision. And we can test now working hypothesis that neuronal circuits perform computations based on the same principles that we see in artificial neural network computations. And this is bringing the field into this direction of using artificial neural network as models of brain functions to test very stringent experimental predictions using these newly available technologies. So I would like to acknowledge the people who did the work, the funding agency, of course, the embassy and the Istituto Italiano di Cultura for the opportunity, and thank you all for your attention.
Thank you very much, Andrea, for also this very interesting and, again, different aspect of neuroscience. Now we can start the panel discussion, so I invite all the panelists to sit on the stage. So first of all, I want to introduce the two additional speakers, and I will ask them also to introduce briefly their own research. So Aurelio Cortese, please.
Okay. So, good afternoon, good evening, everyone. I'm Aurelio Cortese, and I'm a principal investigator at ATR, Computational Neuroscience Labs in the Kyoto area. So today I came from Kyoto. It's a pleasure to be here. In my research group is interested in how people make decisions and how we learn to make maybe better decisions. And within that, what role maybe cognitive functions and sort of like attention or memory or even higher level like consciousness, what impact, what role do they have in the way that we make decisions? In terms of how do we study this, we recruit human participants, and we use a very big magnet, functional magnetic resonance imaging. So we have an MR scanner, and with that we can sort of image how the brain activity changes in response to decisions, to or other maybe perceptual challenges. And we are particularly interested not only in looking how this activity sort of is correlated with choices, but we also try to change this. So to change brain activity to affect behavior and try to find a causal link between activity in the brain and a certain behavior. And for that, we integrate really neuroscience, computational tools, and machine learning or artificial intelligence. And with that, I think I'm good. Thank you.
Thank you, Aurelio. Now, Ludovico Minati from Tokyo Institute of Technology.
Good afternoon, everyone. First of all, thank you, Professor Traversa, for the invitation. It's an honor and a privilege to be here. I currently work at the Tokyo Institute of Technology, Institute of Innovative Research, as a specially appointed associate professor. My background really intermixes three rather different disciplines and ways of thinking: neuroscience, physics, and electronic engineering. I spent many years doing research in neuroscience, particularly functional brain imaging and neurophysiology, sort of connected to what some of you have been discussing today. But more recently, I developed an interest in complex systems. So those properties that seem to come up in similar ways across systems that are actually very different in their nature and in their scale. And in particular, take this approach to design new electronic circuits which replicate some aspects of the activity that we can record in neurons, either at a very small scale or at the scale of the entire brain. So it's a great pleasure for me to be here, and the presentations have been incredibly inspiring, also as regards the interest in combining multiple approaches and multiple disciplines. So thank you again for the invitation.
Thank you very much, Ludovico. So I think I would like to start the conversation. First of all, yes, pointing out that the neuroscience seems to be more and more multidisciplinary. So the more traditional approach is probably the one of Marco, so the biological approach to understanding. And now we are moving to new concepts and the use of computation, somehow the computation of Andrea, which is still at the brain level, or additional tools that can mimic the computation for that. So I would like to ask, maybe I will start with Andrea, what do you think is the practical application then of those approaches? Where are we going with that? Are we going to go to get something that will be useful for people?
Yes, so we have already seen a lot of these kind of trends in the past. So for example, very powerful artificial neural network systems now use approaches and computations that have been directly inspired by what the brain does. So reinforcement learning methods, for example, have been tremendously inspired by the dopaminergic system in the brain. More in my field of research, we see applications, for example, related to the visual world. So nowadays, there's a lot of interest in automation and agents that can autonomously move in the environment using cameras. And so they need to process visual information. And our visual system is incredibly powerful in sort of filtering out noise and not being tricked by elements that instead currently artificial neural networks find very problematic. So these so-called adversarial attacks are a real hazard for society, because even small changes in in pixels of an image could fool a network to think that a panda is a tree. So you can imagine that this can be exploited in very negative ways. You can modify the stop sign in very few pixels, and the car would just think it's like a drive-through. So this is there are very tangible, very objective applications of in transferring our knowledge about the brain system into AI. Not only the other way around, which is what I'm interested in. Yeah.
Thank you, Andrea. Somebody was to continue the discussion of the trend, or Ludovico?
I might pick up briefly from this. I think this is definitely true, and it's interesting to note that it is true not only in terms of how we develop the theory and the software of AI systems, but it's starting to become more and more true in terms of how we develop the hardware of computer systems. Because for the last 20 years, we've witnessed an incredibly powerful growth in computing power, fueled essentially by physics, by advances in device physics that allowed us to have smaller, faster, lower power transistors. But now more and more we are starting to see some unavoidable limitations. And then what to do? Then we are forced to start thinking of doing computation in rather new, unusual ways. So we don't just scale down the way we have been doing computation for 20 years, but we have to look for really totally new approaches. And we know very well that architecturally, digital computers and brains frankly have nothing to do with each other. And one can say that biological brains are amazingly more power efficient and robust than digital computers. So the interesting thing is that this approach is starting to extend to how we design the hardware, and so to involve more and more the engineering aspects, I think.
Thank you, Ludovico. The discussion is shifting a little bit in what computers can do for neuroscience, and instead is what neuroscience can do with computer. I would like to reverse, so maybe Aurelio, what do you think? What computers can do for improving neuroscience?
Yeah, I think I have maybe I have also just an example from our own research, in the sense that first of all, the incredible computing power that now we have, and then coupled with these new advances in machine learning or in artificial intelligence, this allows us to go a little bit deeper in characterizing the brain itself, and then how we can also sort of, by characterizing it better, also how we can then sort of leverage this to have a positive impact in mental disorders. So in our own research, we really try to combine these machine learning approaches to read out a brain state and put people in a sort of closed loop system, so in which they can sort of visualize in a way, or try to really modulate their own brain activity. And then we can do this over time and train the brain to sort of to change maybe a mental state or a behavior. And this can be used, you can think that this can be used, for example, for clinical conditions. You can think of, for example, of PTSD or other disorders in which, for example, you have a very fearful memory that might impact your daily life, or in phobia, for example. Then maybe with these techniques that really pair neuroscience and this really advance both computation and I think characterization, we can then maybe lessen this impact. And this can, I think, can have a really positive impact on society. We are still in the early days, I would say, but I think we're improving fast.
Thank you, Aurelio. I would connect that with the work of Dalila. So are we really going to do gym only in front of computers?
I hope so. No, I'm joking, no. No, it's true. Actually, I was really surprised by what you said also during the introductions. What I see here is like a huge variety of data coming from completely different sources and with very few things in common. For example, what Aurelio Ludovico just said about the artificial intelligence and computational models is something that it's kind of common to all of us, but still in very different ways. And what fascinates me actually from this discussion and from the presentations I heard before are three main points. One, I was thinking, because of the variety of the data we are collecting, I think there is kind of the necessity of standardize. So, for example, I'm thinking of also Aurelio mentioned the patients. When we talk, for example, about neurological patients, the necessity is to find a way to standardize, for example, the symptoms or the assessments or the intervention themselves, which is kind of challenging when you talk about neurological patients because they are so incredibly different and subjective, but it's absolutely necessary. A second point, I think, is the necessity to share the data. Again, because we are talking about the brain and it's so incredibly complicated that it cannot be explained by simply one project or simply one strategy or one domain. So I think it's crucial to share the data coming from all the different perspectives. Personally, I'm very interested recently in the open science and these kind of systems to share actually literally the raw data we collect during our studies. And last but not least, because of the complexity that we are dealing with, it's necessary to, let's say, simplify it, or better, to model them. So I think what Aurelio Ludovico just said about the necessity of computational models and the necessity to render those data in a way that is that they are easy to understand, or they can be approached with within a specific framework and a specific model is absolutely vital.
Thank you, Dalia. Very interesting observations. And I want to connect with the work of Marco now, because we are talking about understanding the brain mechanisms, but Marco is more focused on rescuing for diseases that the brain can have, so motor diseases and kind of things. So how do you think the science is going in this direction? How can you contribute to, we have some hope to solve some of the medical issues that are open, are still open now?
Well,
question, of course.
that will take time, a long time. But I think one thing that we can correct with the sort of general theme that we've been discussing in terms of how we are more and more increasing in the use of quantitative methodology. That is actually something that I see as positive. I started my PhD in 2006, if I remember correctly, and I've seen a transformation in the way we do science, at least in my field, from acquisition of data and very superficial quantification to a really accurate mathematical model. And that has come about because of a change in culture and the way that we quantify things, but it's also a necessity due to the way we perform experiment. Even today in the lab we are doing sequencing, even complex sequencing. There is now an entire field of single-cell sequencing. There is an enormous amount of data from various omics approaches that can't be handled in any other way than expand the use of mathematics. And this will become more and more crucial, especially in the treatment of diseases in the future, because a lot of diseases are based on individualized treatment, and with more targeted, tissue-specific, or even single-cell approaches, we can really have a glimpse of the heterogeneity and the dispersion of the various phenotype in a much more granular way. So having a mathematical model that can handle that, I think, is crucial, also moving for the clinics. And another thing that this has been revolutionary that we are doing in the lab as well is the way of image quantification. So a lot of phenotypes that were previously seen and quantified basically visually, manually, because to this date, I believe the brain is the most powerful computer we have, even for image recognition. Nevertheless, we are now experimenting with a variety of neural networks and suprapixel and whatnot to try to really quantify complex morphologies that we are dealing with our imaging. And that has made an enormous difference in our ability to interpret the data. So I do think it's important. And I think that's a direction that will be productive. Will not generate cure tomorrow, but maybe, who knows, in a reasonable time, it will be beneficial.
Thank you very much, Marco. I agree that all science, not only the neuroscience, is very much changed in the last years, and that big part of changes is due to mathematical modeling that becoming not only for understanding the results, but also for predicting the results. And so the predictive also power of mathematical methods is extremely important. So if anybody wants to say something before closing.
I think that one aspect that is interesting to highlight is this. So multiple branches of science collaborating together, of course, is nothing new, right? Physics has always been very instrumental to biology and certainly to neuroscience. But I personally think what is particularly interesting to observe over the course of the last 10 years is that the way various disciplines interact with each other is becoming much deeper, much more intertwined, and much more conceptual. So it is no longer a case of computer science, for example, offering tools to neuroscientists and neuroscience offering some inspiration to computer science, but they really start co-evolving, and there are more and more and more people who can't really define themselves as belonging to one field or the other, but they define themselves as belonging to the intersection of these fields. And I think this is one of the most important paradigmatic change that we are observing everywhere, and it is actually what links together all the presentations that we have seen today. And this is one aspect which I think we should take home from this meeting.
Totally agree.
If I may add to this comment, I think actually there's an even deeper problem if you want to call it problem, but in this observation, that is the traditional role of the single lab that is doing a specific type of research is now sort of jeopardized a little bit. It's very difficult to have a lab that is focused only on a specific aspect, at least in neuroscience, of the research. You need to integrate from many different approaches, and that means that you need to interact with other labs. So you see more and more that labs tend to merge with other labs in terms of resources, but also efforts. And it seems like we're going a little bit more in the direction that we see in physics, where, for example, in particle physics, you have hundreds, if not thousands of labs, of course, consortia of labs that join efforts in order to advance science. And I think in neuroscience, we're going to see something similar. You cannot have in a single lab all the necessary expertise to tackle a given problem from different angle as it is necessary nowadays.
Thank you very much. So for sake of time, I'm closing the panel discussion. That was very interesting. So I want to thank all the audience, the participants, and especially our speakers again, Ludovico, Aurelio, Andrea, Dalila, and Marco, for their fantastic work, and just continue to do like that. Thank you very much.